Dear guests, dear friends, it's very hard to be interesting after this beautiful lecture, so I will be very short, really just take the opportunity to introduce quickly our laboratory, what we call Epigenetics and Chromatin Research Laboratory. It is located partly at the University of Szeged in the Department of Biochemistry and Molecular Biology, and as well in the BRC in the Institute of Biochemistry. And the reason I'm introducing it to lead to the presentation of Petra Szili, who is a senior student at this laboratory. So it has been mentioned several times today, and again, I'm sure everybody knows it, that the cornerstone of our modern biology knowledge is that DNA directs RNA synthesis and RNA directs the synthesis of proteins, and the action of proteins will determine the different features from simple one as, if we might say, simple, how the cell will divide and what the cell will metabolize, what will produce, how flies will show up on the coral of their eye or in the shape of their wings, which can be very easily studied, and very complex, how humans will develop from a single cell to mature adults. All these is encoded in the DNA, and over the second half of the last century, we learned many, many things, how these processes are regulated and how the DNA, the information which is encoded in the DNA leads to these beautiful phenomena, what we can see. Over the last decade or 15 years, however, we are realizing more and more that the process is much more complicated even, and there are other layers of complexity in which partly arise from the environment. And in this sense, environment means everything what surrounds us and what surrounds our genes and our proteins within the cell, the food what we are eating, and even the personal experiences what we experience in our childhood. These environmental effects will seen in differences. For example, completely identical twins who have the same genetic makeup, and even so their looks differently and they have very different features, or in the action of genes which supposed to determine one particular color, for example in the eye of the fly, but will show some variegation, it is called, or in the growth of cells which instead of growing normally became cancerous. All this is done by the environmental effect, what we altogether combine in the term epigenetics. Epigenetics. The epigenetics factors altogether is believed to constitute an epigenetic code, which has mainly two components. One, which is the methylation of DNA, or in general modifications on DNA, and modifications of the proteins which serve to pack the DNA into the chromatin structure so as it could fit in the tight environment of the nucleus, and in the meantime it will determine whether the genes, the information which is encoded in the nucleotide sequence can be read or cannot. The genes can be activated or will be silenced in a particular situation. We are particularly interested about this structural unit of the chromatin, which is called the nucleosome, in which a so-called histone octamer forms the center around which the DNA is wrapped. And we are studying this in different model systems using flies, since easy to do genetics on them, and human cells, since these are obviously closer to the everyday problems what we would like to solve. And we use the tools of genetics, the cell biology, and the biochemistry as well to explain how the changes of the chromatin structure, and particularly on the modifications on the histones, among them ubiquitination, will change the expression of the genes, and as a result of the gene expression changes, how it will affect the phenotype of an organism, of a person. And into this work joined us Petra, who is a first-year senior student, and in biology she is a second-year BSc student. She had numerous previous awards, among them an international certificate of award from a biology Olympiad. She participated in it, and as you can see, she's well dressed and ready to climb up the high mountains of the science and make discoveries. We are particularly proud to introduce her as the support in our laboratory to the numerous other supports what we are receiving from different organizations. So I asked Petra to introduce her project, which is a completely new one, but obviously relates to the epigenetic studies what we are performing.
I want to thank Professor Boros for his introduction, and in the next minutes I want to talk about the work I've done in the last couple of months, which was mainly about what happens to cancer cells when their drug resistance change. So, one of the greatest medical challenges of modern mankind is the diverse group of diseases called malignant neoplasm, or more commonly cancer. Cancer takes almost 8 million lives every year, and the number of patients affected by the disease is currently increasing. In Hungary, actually every fourth person dies in cancer, so it's the second most common cause of death. One of the problems with cancer is that the cost of medication is really high, so it's both our personal and economical interest that we should make the treatments as effective as possible. One of the traditional approaches in treating cancer is the use of chemotherapy, when the malignant cells try to be killed by the use of cytotoxic drugs. But sadly, on some of the malignant cells, these drugs has no effect at all. This phenomenon called drug resistance, and it can be natural or acquired. We talk about natural drug resistance when the cancer cells prove to be resistant from the beginning of our treatment. But we talk about acquired resistance when the cancer cells are originally sensitive and they become resistant during our drug treatment. One of the most common causes of acquired drug resistance is the overexpression of ABC type transporters, which is a transmembrane pump family, and they can export drug molecules from the cells. One of the most abundant type of ABC transporters is the multi-drug resistance 1 protein, or shortly MDR1. MDR1 has 2 transmembrane and 2 ATP binding subunits, and on the intracellular side of the cell it can bind ATP, and with the energy of ATP hydrolyzation it can change its conformation and pump out the drug molecules from the intracellular to the extracellular side of the cell. MDR1 is a popular theme for research in molecular biology because if we can find a way to manipulate the cell's ability to express MDR1, then we can probably find a new therapeutic target for cancer. Our collaborators, the group of Gergely Szakács, are currently working on this matter. They used an originally sensitive cancer cell line and they treated it with doxorubicin, from which the cell lines became multi-drug resistant. After that, they used another type of drug treatment, from which these cells lost their drug resistance. They became sensitive again, which we found particularly very interesting. And after that, they treated these cells with doxorubicin again, and the sensitive cell lines became drug resistant again, so the loss of drug resistance proved to be reversible too. So our question in my research was that what is the cause of this reversible drug resistant change? What are the main regulatory differences between the cell lines? The first question, I wanted to know if the change in drug resistance is caused by the change in ABC transporter activity. So I used doxorubicin, which is a fluorescent ABC transporter substrate, and treated the cells with it. So I expected that in the resistant cell lines, the fluorescence rate is going to be lower because they can pump out these drugs from them. But in the sensitive cells, the fluorescence rate is going to be higher because they keep the drugs in them. So I performed effects analysis, which can sort the cells based on the rate of the fluorescence, and found that in the resistant cell lines, the rate of the fluorescence was more than 10 times higher than in the sensitive cell lines. So I saw it proved that this is because the change in activity of ABC transporters. As we all may be well aware of it, the information in the cells flows through DNA, mRNA, and protein before it finally materializes as function. The process can be altered at practically every step with gene amplification and the change in transcriptional regulation between DNA and mRNA, with RNA processing, control, RNA degradation, or RNA transport and localization change, or the change in transcriptional regulation between mRNA and protein, and the change in protein degradation or protein activity regulation between protein and function. So, because I witnessed the change in cell function in my research, I decided to go backwards in the flow of the information to find out at which step the system was mainly altered. At first, I wanted to know if the level of MDR1 protein changed in these cell lines, because it's the most abundant ABC type of transporter. So I performed the Western blot, and I found that in the resistant cell lines, the MDR1 protein is present in a much higher level. One of the most initial ways of regulating protein level of the cell is to regulate the mRNA level from which it was translated. So as a next step, I wanted to know if the MDR1 mRNA level also changed in the cell lines. So I performed the qPCR on MDR1 cDNA and found that compared to the sensitive cell lines, in the resistant cell line, the MDR1 mRNA was present in 100 times higher level. As a next step, I wanted to know if the reason for the change in MDR1 level can be gene amplification, because it's a very common case in cancer cells since their genome is highly unstable, so they can lose or gain genes during their lives. So I performed the qPCR analysis on MDR1 gene exon 7 and found that this small change in copy number can't explain the high change in MDR1 mRNA level. So as an alternative explanation, I had to know that what happened in the transcriptional regulation. The structure of the DNA without any modification is very tight, so it's hardly available for transcriptional factors. Excuse me. and if we want a higher transcriptional activity, then we need to make the DNA structure loose up. And this can happen with the modification of histone proteins, for example, the acetylation of histones. So in my last experiment, I wanted to know if I can find any histone acetylation change between these cell lines. So I performed the Western blot analysis with two types of histone antibodies, H3K4, H3K18 and H4K5. Excuse me. And I found that in the resistant cell lines, the level of acetylated histones seems to be mildly higher. So the acetylation of histones may be increased in the resistant cell lines, but I need more data about this field to be sure about that. So as a summary, I witnessed the change in cell function as the cells became drug resistant. I saw that this is caused by the change in MDR1 protein level change. And I saw that the mRNA level was also changed, but I saw no evidence of gene amplification happening. So I had to reconsider that maybe transcriptional regulation was changed in the cell lines. And I checked practically one type, the histone acetylation. In the future, I want to have more detail about how the histone acetylation changed the phenotype of the cells and how these acetylated histones distributed on the MDR1 gene. And after that, I want to know more about other types of transcriptional regulation, other types of histone modification, and for example, the binding of transcriptional factors. I want to thank my mentor, Professor Imre Boros, and all the staff members of Epigenetic and Chromatin Research Laboratory and the Department of Biochemistry and Molecular Biology for their generous help and support during my work. Thank you for your attention.